I'm lucky to have to work with her in some few smaller projects. So I know she will have a very interesting presentation about algae in circular economy and also about growing algae on digestate, which is also algae interest. Please, Carol.
Thank you, Marja. I'll try and keep to time, but do stop me. Thank you so much for inviting me along today. It's been a real pleasure to listen to everybody's talks. And I think you'll see as I go through how the work that we've been doing in Northwest Europe fits into like nearly everybody that's been talking today. So I'm going to focus on microalgae grown on digestate from food processing facilities. I'm going to give you like 10 minutes is barely enough time. So I'm basically going to give you an overview and point you in direction of finding further resources because the project's finished and there's quite a few outputs. So the real aims of this 𝐜𝐢𝐜𝐵 project that was funded by European funding, Interreg funding rather than Horizon funding. So it was really about working with industrial partners. And we wanted to take digestate from anaerobic digesters and develop new market opportunities. And the idea was that the digestate coming out of anaerobic digesters is used as fertilizers, but there's often a problem of having too much digestate, and there's a possibility of runoff from agricultural lands into water systems, causing eutrophication, which causes huge pollution problems. So we wanted to use that digestate to create new market opportunities and turn that liquid digestate into biomass that could be used as sustainable animal feed, both in terms of feeding animals and also in aquaculture. And particularly looking to produce new source of protein for, as I said, animal and fish feed, but also for other high value products. So this is basically how the project fitted into a circular economy. We took manure and agricultural waste that are fed into anaerobic digesters. So this was, we worked with industrial sites and with research institutes that were already doing work on anaerobic digestion. So we took the digestate, which is very rich in nitrogen and phosphorus. We fed that to microalgae. We then did analysis and extracted the high value molecules and also the protein for animal feed. And then of course the waste from animal feed goes back into the anaerobic digestions, therefore creating a loop and a circular economy. So a bit more detail about the different work packages of the project. So this is basically the same circular loop. We had the work package 1, it basically consisted of 3 pilot facilities, one in the UK, one in France and one in Belgium. I'll talk a little bit more about those. So they were basically pilots or demonstration facilities that were built close to or using anaerobic digestate as shown here. And basically the first work package involved setting up those facilities and then operating them over a period of several years. Work package 2 consisted of characterizing the biomass that was produced from the algal facilities and then developing that algal biomass into products that could be used commercially. And as part of the main project, we did piglet feed trials. We then got additional funding, which we called a capitalization work package. Well, that's what we were told to call it from Interreg, which we did some fish trials with. So I'll talk a little bit about those in the amount of time that I've got. Another big work package was to take all of the information and look more widely across Europe to develop tools that people that wanted to use this technology could use. So we mapped where anaerobic digestion facilities were, where there was a big need, where there were problems with agricultural runoff, and we developed a whole series of tools that are still available, and I'll point you in the direction of that. And then that was really worked with long-term rollout, sort of moving forward, how could we take the technology forward? So of course, there were a huge team involved in this, and I actually did very little of the work. I just kind of oversaw it and got the project off the ground to begin with. But the project was led by Swansea University, which is where I work. We worked with an industrial facility based down in Devon, which is where my hometown is, called 𝐜𝐢𝐜𝐫𝐜. We used their facilities, and also in the UK, Birmingham City University were involved in the developing all of the decision support tools. Then in France, there were a number of partners, again, research organisations and university, working with a large industrial company, 𝐜𝐢𝐫𝐿, who are as a major company in terms of developing pig feed products, or pork and associated meat products. Also, ANSYS, who undertook all the microbiological and toxicological analysis for us and fed back into advising the best way to process and to use the biomass, making sure that the waste we used was acceptable in industry. And also ACA3, this agriculture, were involved in the long-term rollout of the project. Then Belgium, we had the University of Ghent and InnoLab, who were experts in characterising digestate. And then KIT worked together with Birmingham City University in terms of decision support tools. We had a big advisory board that helped us move forward on the project as well. So basically, a little bit about the three facilities. This was the facility based in the UK, in Plymouth, working with 𝐜𝐢𝐜𝐫𝐜. And then the French partners based in Brittany had their site at this industrial facility in 𝐜𝐢𝐫𝐿 in Lamballe. And then the third one in a lab working with Ghent. So we were all using slightly different microalgae. In the UK, we were using chlorella and cenodesmus. We did different runs using these as monocultures, and we used them both in the autotrophic mode and mixotrophic, where we fed them a source of carbon. And I'll talk a little bit more about that if I've got time. The French partners used heterotrophic species, Autocritrium, which is very similar to Schizotrium. That's very, very rich in fatty acids. And then Ghent used a mixed culture of chlorella and desmodesmus. So a little bit more now about the UK facility.
2 minutes.
Say that again.
2 minutes.
Is that all? Yeah, 10 minutes goes so quickly. Oh, it's ridiculous. Okay, so for the UK facility, we used sunlight, but we also used a source of carbon, and we used membrane technology to concentrate the algae up and then grow them in mixotrophic mode before we developed the biomass to feed for animals. So we used a combination. I want to say that we not only did the large scale trials, but we did a lot of laboratory analysis here, just showing you some photos, and then lots of different plots of the analysis that we did. We did detailed analysis of the microalgae that we produced. And one take home message that I want to put across is that algae can be very, very varied in their the concentrations of proteins and lipids. They vary depending on how they grow. So we did detailed analysis of all of the main components using the digestate and the algae grown in a control media as well. And we particularly focused on fatty acids, looking at the changes in the fatty acids, both in the algae but also in the fish feed. So I just want to show you here some differences. So we actually analysed the tilapia steak when we did fish feed trials, and we found that the fish flesh contained much better concentrations of PUFAs and protein when the fish were fed on the digestate than when they were fed on a standard feed. I want to point you in the direction of work package 3 so that you can go to this website here, 𝐜𝐢𝐜𝐷ST, and find out all about the decision support tool. So you can search if you wanted to set up a business, you could search where the AD plants are, You could search various staff, stakeholders, and determine exactly how much biomass you can use linked to the waste that you're producing. As part of the project, we set up a big exhibition in a Durvale experimental farm. And again, I recommend that you look at this YouTube video to find out all about that. We published a whole series of papers, which you can find on our website, and a whole series of output reports that are useful for the whole community, not just for this project, but lots of best practice guidelines and recommendations across all aspects of the project. So please do tap into this huge amount of information that is available out there from the project. And I think all of these slides are going to be available, so the links are there. So that basically I've been able to give you a very short introduction to the project in the amount of time that I've had. Thank you very much.